Hello first-year students, I'm Kathleen Morrill, a student in Eleanor Carlson's lab. And in this lecture, I want to help us bridge the gap between probability and statistics by introducing probability distributions. Distributions help us answer more involved questions about probability, such as the probability of an outcome greater than, less than, or between values. These type of questions would quickly become tedious to answer via those tree diagrams or probability formulas. Probability distributions will also be crucial for statistical testing when we want to draw inferences from our samples. We'll be going through why we use distributions, as well as a variety of examples that can be applied to different scenarios and experiments. So let's consider the following scenario. You are a student interested in gene regulation. You've read some previous studies that show that a certain protein, called alpha, binds to DNA and serves some important function in the cell. You first want to know, where does it bind, and what are its protein binding sites? So you perform an experiment in which you stabilize those protein-DNA interactions. You chop up the DNA into segments through digestion or shearing, and you apply an antibody against alpha to pull it down, and with it, whatever DNA is bound. You sequence this DNA, obtaining sequencing reads, which you subsequently map to the reference genome. You may already be familiar with this type of experiment. It's called the ChIP-seq experiment, and it involves immunoprecipitation and sequencing of protein-bound DNA. In an ideal world, those sequencing reads would pile up approximately at the locations of the protein binding sites, and it would be clear beyond any reasonable doubt where to map those sites. However, in reality, your mapped reads are much more likely to look like this result. Does that mean that alpha is binding everywhere? Well, no, not really. There are a lot of sources of noise in biological experiments that could explain this. But can we still identify the protein binding sites? Sure, by eye, we could deduce some protein binding sites among the noise. Some sites might be a bit more ambiguous, and you might end up missing sites or identifying the wrong sites altogether. So how can we tell an actual site from a random accumulation of sequencing reads? Well, say we divided the genome up and binned the number of sequencing reads that overlap each region. Do we have a better sense now for where reads pile up more than random? Is it a site where there are 10, 12, or 15 reads? We want to know how many reads are there on average and how much do they vary? What is our probability of observing a high count region just by chance and not by actual biology? What we really need here is a better sense for how these counts distribute across the genome. A distribution is a function describing the values assumed by a random variable. This here is a histogram of the sample, showing the frequency of sequencing read bins. Here we can see that the majority of genomic regions in our experiment don't really exceed 5 to 6 sequencing reads. It's rarer than even to observe a region that has greater than 7 reads. And that already tells us a lot about the baseline noise. But our observations do not tell us where to draw the line for protein binding sites with any sort of confidence. Now, if we had a theoretical expectation to describe the probability of seeing these read pileups, we might have a better sense for what observations are rare by chance. In this imaginary probability distribution, which describes the probability of observing X numbers of mapped reads in a region, we can see what counts are expected under 5% of the time. A random variable is the numerical description of the outcomes of an experiment. Less formally, this is whatever you're measuring or sampling. A random variable can be discrete or countable, a value from a finite set or infinite set of integers, like the number of heads you flip on a coin toss, or the number of copies of a genetic allele. Or a random variable might be continuous, assuming any value in an interval, such as human height. All of the values that a random variable can assume are described by its sample space. For a series of 4 coin tosses, for example, you might observe 0, 1, 2, 3, or 4 heads. For a genetic allele, you might be observing none, 1, or 2 copies. For a range of heights, The random variable may assume any value within that range. So probability distributions are mathematical functions that describe the probability of values assumed by a random variable. As is one of our core probability principles, the sum of probability across all values will equal out to 1, accounting for 100% of the probability. Probability distributions come in two flavors, discrete and continuous, reflecting the sample space of the random variable. There are many theoretical distributions, and we are going to cover a handful of use cases that can also happen to be very common in biology. Which distribution to use really comes down to the experiment, its parameters, and what you're measuring. All these theoretical distributions have some mathematical function that takes in parameters and outputs probability of observing a given value. But for the most part, we'll not be calculating these results by hand, and we'll instead be using functions that have been implemented in R. You'll get to practice this in the distributions workshop. Discrete distributions are probability functions over a countable sample space. First, let's take a look at the uniform distribution. This is the simplest discrete distribution in the books, and you're already applying it with your probability exercises. Think about a coin toss. There are two possible outcomes. Each are equally likely, so the probability distribution is just 50% heads and 50% tails. Or consider rolling a well-balanced die. You'd expect the probability distribution to be uniform, with each probability being 1 divided by the number of sides. This 8-sided die has 8 possible outcomes, each with a probability of 12.5%. But what about an experiment where you only consider 2 outcomes that aren't necessarily of equal likelihood? This would be the Bernoulli distribution. Here, the random variable is binary, success or failure, yes or no, true or false. We can even take the dice rolling experiment, and if we consider rolling an 8 to be our success and all other rolls to be a failure, then our probability distribution would look like this, where the probability of getting 8 is a lot lower than the probability of getting not 8. How about those exercises where you calculated the probability of getting 3 heads in a series of 6 coin flips? That's described by the binomial distribution. It's a series of Bernoulli trials. For a sequence of n trials with 2 outcomes, success or failure, and constant probability of success p, the binomial distribution describes the probability for getting x number of successes. The binomial distribution for a coin flip, therefore, with a 50% chance of getting the desired heads, is very central. Most of our probability is concentrated around getting 3 heads. Say I ask you to calculate the probability of getting 3 heads on a series of 6 coin tosses. Well, right now you might start drawing out your tree diagram, trial by trial, tediously multiplying and summing up the probabilities of each combination of 3 heads. But really, you don't have to do that. This is a binomial function, so we can go into R and apply the binomial function. We want the probability of 3 heads. We have 6 trials, and the probability of getting heads is 50%. So bam, the probability of getting 3 heads is 31%. You don't have to do tree diagrams for this. Back to the binomial distribution. If you're like me, you spend a lot of your free time rolling dice and hoping for the highest value. What's your probability of getting the highest score, 8, on 6 rolls of an 8-sided die? That's binomial too. It's just now that the probability of success is quite smaller. And now the distribution gets skewed towards getting none or very few successes. Likewise, we can describe the probability function for failures before getting a certain number of successes. This is the negative binomial function, and it maps the probability of reaching a given number of n successes over endless Bernoulli trials, put in terms of how many failures will occur. For all the distributions based around trials, we've been assuming independent events. In our probability exercises, however, we've seen examples of the classic ball drawing experiment, in which a population is sampled without replacement. Here we have a population of green and blue balls. And the probability of success here, defined as drawing green balls, changes with each sampling event. Starting at 33.3% for our first draw, drops down to 31.8%. Then the next sample changes the probability once again. We have a probability distribution for this situation too. It's called the hypergeometric distribution. The parameters here reflect the population size, the number of successful entities that might be drawn, so here the number of green balls, and the constant sample size that we're taking with each draw. Let's consider another scenario. How about if we are observing a biological event that occurs over a time period? The Poisson distribution is like the binomial distribution in that it's discretely describing the number of successes, but instead of over a set number of trials, it's over a given interval. For example, how many cell divisions occur over the course of an hour? The events occur randomly, and Poisson distribution assumes a set constant rate of occurrence, lambda. Here, lambda is 2 divisions per hour, and we see that our probability is mostly centered around getting 3 or 4 cell divisions over the course of 2 hours. Back to our protein binding experiment. Say we know that protein alpha binds to 5% of the genome. We sample 20 random locations on the DNA, and we want to know our probability of observing at least 4 binding sites. Well, this is essentially a binomial distribution. We have 20 trials, that is, samples, and we have discrete sample space, success, binding site, or failure, no binding site. We can construct the distribution and add up the probabilities of each number of binding sites from 4 to 20, or we could add up the probabilities of getting 0, 1, 2, or 3 binding sites and subtract that from 1. Therefore, our probability of observing at least 4 sites is 1.6%. All of the discrete distributions we've gone over so far have been in the form that answers the question, what is our probability of observing each value for our random variable? The probability mass function, or PMF, is your distribution of probability for the random variable equaling a value. The cumulative distribution function, or CDF, reflects the probability of obtaining X or less. It is always increasing until it reaches 100%. Its complement, the complementary CDF, reflects the probability of X or greater and is always decreasing, approaching 0%. And as you see here, that 1.6% probability of observing at least 4 protein binding sites is reflected right on this plot. In summary, the probability distribution you apply really depends on the question you're asking. You can be flipping a coin and just asking, what's the probability of heads and what is the probability of tails? And you'd be describing the uniform distribution. Now, if you consider heads to be a success and tails to be a failure, your probability mass function is exactly the same, but now you're describing a Bernoulli trial. Repeat that Bernoulli trial multiple times, asking the probability of achieving a certain number of heads. Well, now what you're asking for is the binomial distribution. And if you want the probability of at least X heads or at most X heads, you'd want to construct those cumulative distributions, and so on. So it's all about defining your experiment, your parameters, and what probability you're modeling. Let's go through an example. My friend and I are running a dog spotting competition. I tend to see 5 dogs a day during waking hours, and my friend averages on 2 dogs. Today we decided that if we both see 4 dogs or more, we'll go out for drinks. Whoever sees more dogs buys, and if we fail, well, then we stay in to study. What type of probability distribution applies to this scenario? Well, for one, it's discrete. It's the number of dogs observed. It's over a time period, so let's go with Poisson. And I'm asking about the odds of 4 dogs or more. So I want to construct a cumulative distribution. In fact, I want the complement form that reflects a greater than 4 question. These are our respective Poisson probability mass functions. And here are our respective Poisson complement cumulative functions. Assuming the dogs I see and the dogs that they see are independent events, I can use our functions for the Poisson distribution to calculate my and their cumulative probabilities. What I want here is to subtract the cumulative probability of observing 3 dogs or less from 1 to obtain my probability of seeing 4 dogs or more. And do this the same for my friend, except now their average rate of dogs that they see per day is 2. Then I can just multiply our two probabilities and obtain a probability for us both seeing 4 dogs or more. And it looks like our odds of going out tonight stand at a measly 10%. Continuous distributions are those probability functions spanning a continuous range of values for the random variable. Geometrically and numerically, continuous functions are interesting and sort of weird. The probability evaluated at a given point on the range of values for the random variable is 0. Here, at x equals 1.2, the probability of that exact value, 1.200000000 at infinitum, is nothing at all. The same for 1.2001 or any other value on the range. But at a given interval, we can evaluate the probability of observing any value within a given interval. And that ends up just being the area under this curve, or the integral of our probability function. The uniform distribution's back again, except this time, probability is uniform over a range of values that the random variable assumes. Consider, for example, a spinning top. The angle it comes to rest at is random, and all angles are equally likely. So this is basically just a line across our range. Like with discrete distributions, we can define the probability function, for continuous distributions, the probability density function, or PDF, as well as its cumulative version to evaluate probabilities of less than or equal to a given value, or the reverse, by subtracting that probability from 100%. The distribution of time between events in a Poisson process is a continuous probability distribution that we call the exponential distribution. In the same scenario we had for the discrete Poisson distribution, the exponential distribution of time between cell divisions would look something like this. Likewise, if we wanted to know the probability of a cell dividing in the first 30 minutes since the last cell division, then we can apply the cumulative exponential function in R, giving it 30 minutes or less at a rate of 2 cell divisions per 60 minutes. We see that the probability of observing another cell division in the first 30 minutes since last time is 63%. The normal distribution is the most widely applied continuous distribution. It's also known as the bell curve. It takes in 2 parameters, mean and standard deviation. It's symmetrical around its mean, and it can be applied to many natural phenomena. The standard form of the normal distribution strictly has a mean of 0 and a standard deviation of 1. Here we have the normal distribution of male height, where the mean is 70 inches and the standard deviation is 3 inches. Another interesting feature of the normal distribution is the way that its probability density distributes. Within 1 standard deviation of the mean value is contained 68% of the probability. Likewise, cumulatively, 95% of the probability of observing values are for values within 2 standard deviations of the mean. That means that on the very tails of the distribution, outside 2 standard deviations, those values only have a cumulative probability of 5%. The mean, of course, is the average of the values for the random variable. We do distinguish the mean of the population as a whole from the mean of a sample drawn from the population, giving these different symbols. Likewise, the standard deviation is a measure of variation among those values, or dispersion. Here's an example. If we have a solution whose pH follows a normal distribution, and we prepare 5 beakers of the solution, then we can calculate the sample mean and the sample standard deviation. Then we can model a normal distribution using these parameters, the sample mean and the sample SD, to estimate the probability of observing a pH greater than or equal to 3. The z-score transformation is a normalization method that places values x of the random variable in terms of standard deviations from the population mean. If I tell you that the true mean and standard deviation of the solution are 1.8 and 0.4 respectively, then we can calculate the z-scores of our observed values in this manner. The whole point of these theoretical probability distributions is that they allow us to make inferences based on our sampling of a population's distribution. And that's what we call statistics. Say we have this population of blue spheres that vary in shade. Each time we're taking a sample, we're calculating what the mean shade of the spheres in each sample is. And as we do so, taking subsequent samples, we are building a sampling distribution. We can construct a sampling distribution by repeated random sampling from a population and calculating the sample means. Here we grab our first sample, calculate the mean, grab additional samples of the same size, and continue to do so until we build up a sampling distribution. And this sampling distribution will largely be centered around the true mean of the population. And while the population's distribution may not be normal, often a distribution of sampling means will center around the true population mean. And in fact, the standard deviation of this sampling distribution is what we refer to as standard error, or standard error of the mean. The central limit theorem, therefore, asserts that the sampling distribution will approximate a normal distribution centered around the true population mean. However, the CLT only applies to samples that are sufficiently large. What's large? Well, the magic number for large is often quoted to be 30. In a sense, this is arbitrary, but it isn't exactly. What about when the sample sizes are not quote-unquote large, when the standard deviation can't be approximated? Well, it's then that we need the t-distribution. And the population standard deviation is unknown, and you're trying to estimate the mean of a normally distributed population. The distribution that fits best is the student's t-distribution. The student's t-distribution plots the t-scores of each small sample from a normally distributed population. That is, the values are put in terms of the sample's standard deviations. At a certain point, the samples are sufficiently large enough to approximate a normal distribution. That tends to happen at samples of 30 or more. It's then that the t-distribution begins to truly look like the normal distribution, and it makes more sense to assert the standard deviation use z-scores. The biggest difference between t-scores and z-scores is this squared sample size term applied to the sample standard deviation, substituting in for the population standard deviation. Another highly used distribution would be the chi-squared distribution. It's used to compare observed values to the theoretical normal distribution. And here we're defining a really artificial random variable that comes from a hypothetical sampling of the standard normal distribution. That is the one where the mean is 0 and the standard deviation is 1. The parameter here is degrees of freedom, or the number of samples drawn from the normal distribution. When the degrees of freedom is 1, we are supposing that the normal distribution is sampled only once. Most often, the sampled value will be 0 or close to 0, since most of the probability density is towards 0 on the normal distribution. Ergo, the square of 0, or a very small number, is also 0, so most of our probability density in the chi-squared distribution is also concentrated around 0. When we bump this up to 2 samples, we're now adding together the squared values drawn together. This is still likely to be small and close to 0, so the chi-squared distribution still is kind of concentrated around 0 or 1. But as soon as we go up to 3 degrees of freedom, the chances that those 3 squared values add up to more than 0 is higher. The chi-squared distribution also has 2 properties. The mean of its distribution is equal to the number of degrees of freedom, and its variance is also just 2 times the degrees of freedom. We'll cover the chi-squared distribution more extensively in parameters and statistical testing, but just know that the shape of this distribution changes dramatically as more samples are taken. We can also now define a chi-squared statistic. The chi-squared statistic puts the observed values x in terms of the mean or expected value. It's just x minus the mean value squared divided by the mean value. And the sum across values follows the chi-squared distribution. Let's do an example. The relative expression values of three genes, G1, G2, and G3, are all equal to 10 TPM, or transcripts per million. We think that if we knock down this other gene, H, that the expression of these genes will change. After we do our experiment, we observe TPM values of 9, 11, and 11. Can we conclude that H knockdown has changed the expression level of these genes? What we can do is calculate the chi-squared statistic for each of these genes. Their expected value is 10, and their observed values from our experiment are 9, 11, and 11, but the chi-squared values all come out to 0.1. Because the sum of these chi-squared values follows a chi-squared distribution, we can apply the chi-squared distribution in R for observing a total sum of squares of 0.3. Our degrees of freedom is 2 because we have 3 samples. And we see that the probability of observing this value, just by chance of drawing from the chi-squared distribution, is 86%. So we cannot conclude that the knockdown of H changed the expression levels of the G's. It could have easily happened by chance. So to conclude, I know this has been a lot of different probability distributions. We have our discrete distributions, starting with the uniform distribution, in which case all values the random variable can assume have equal probability. We have the Bernoulli trials, where you have only 2 possible outcomes for your random variable, success or failure, and they might not have equal probability. We have the binomial distribution, in which it's a series of Bernoulli trials, independent trials that don't affect each other's probability, so the probability stays constant, and it measures as the random variable the number of successes. Likewise, we have the negative binomial that puts this in terms of the number of failures before a given number of successes. And we have, when we sample from a population without replacement, and the probability is constantly changing with samples taken, the hypergeometric distribution. Finally, for the discrete distributions, we have something that's not in terms of trials or samples at all, but over a time period is the Poisson distribution, the number of successes that occur in a given time period. For the continuous probability distributions, this is over a continuous sample space where the probability at any given value that the random variable can assume is 0, but we can calculate the probability over an interval of values. We have, of course, the uniform distribution again, this time over a continuous sample space. The exponential distribution, which is its continuous cousin of the Poisson discrete distribution, demonstrating to us the probability of a given time period between occurrences of an event. And then the normal distribution, the bell curve that describes a lot of natural phenomenon, described by 2 parameters, the mean and standard deviation, as well as 2 continuous sampling type distributions. Both the student's t and chi-squared distributions are parameterized by degrees of freedom, or experimental sample sizes, and are used extensively in statistical testing. That concludes the distributions I wanted to cover in this lecture. I'll have these exercises posted also on the Bootcamp website as well. Remember to go and take the quiz after you feel comfortable with the material, before the live session. Finally, I'd like to give a little preview of the live discussion we'll have for this lecture. I'm a student from Eleanor Carlson's laboratory. We do a lot of bioinformatics and genomics, and what makes our research unique is our comparative and evolutionary biology approach. My thesis project focuses on the behavioral genomics of the domestic dog, so I'll be sharing how I explore the data we've collected from thousands of pet dogs using distributions.